This video covers Advanced Higher Biology, Unit 1, Cells and Proteins. Key Area 1, Laboratory Techniques for Biologists. Specifically, today we're going to look at the health and safety, so section A of that key area. We'll define a hazard, give examples of possible hazards in the lab, define a risk, give the purpose of a risk assessment, and describe some control measures we might use. So a hazard is anything in a lab which could potentially cause harm. Common hazards within a lab include things like toxic or corrosive chemicals. Any toxic hazard should be labelled with the famous skull and crossbones symbol. Corrosive chemicals are labelled with a tipping test tube and refers to any chemical likely to cause a high degree of corrosion, which is another word for rusting. Hot objects can have a hazard symbol with 3 wavy lines rising from the base. Sometimes there's a hand above it, and often in a lab it will just have a sign that says warning, hot or burn hazard, do not touch. Flammable symbols you'll probably have seen around. Flammable substances tend to have a fire symbol and are likely to go on fire if given a source of ignition. Organisms which cause disease, so-called pathogenic organisms, have the symbol which is often linked in popular culture to biohazards. Mechanical equipment can have many different symbols, usually displaying quite unpleasant outcomes if you're to become injured. So here we see a sharp object going to hand, or a person's hand going in between several rotating screws. Often there's many safety precautions put in place to prevent these things from happening. The risk is the likelihood of a harm arising from exposure to a hazard. Every time a new piece of equipment is brought into a lab or a new chemical ordered or technique carried out, the overall safety is assessed using a risk assessment. In a risk assessment, the likelihood of a harm is judged for the new hazard and is given a rating, so low, medium, high. So if it's unlikely that a piece of equipment will cause damage or harm, it's given a low rating. If it's likely that it could cause a high degree of harm, it's given a high rating. The risk of that harm actually happening is assessed giving a second rating. So this is the level of risk. The two ratings are considered together. Based on this, procedures are put into place to try and avoid the hazard and reduce the risk. This is what we call control measures. So the hazard is the harm that could be caused, and the risk is how likely it is to happen. We have control measures in a lab. This includes everything from appropriate handling techniques, protective clothing and PPE, equipment, and aseptic techniques. We should also consider using the smallest affected masses, volumes, and concentrations of dangerous chemicals and limiting the length of exposure we have to that hazard. It's quite a short video. We should now know that hazard is something with the potential to cause harm. We should know that this includes toxic or corrosive chemicals, heat or flammable substances, pathogenic organisms, and mechanical equipment. We should be able to define the risk as the likelihood of harm arising from exposure to a hazard, and that a risk assessment involves identifying control measures to minimize this risk. The control measures can include appropriate handling techniques, protective clothing, and equipment or aseptic techniques.